Welcome to the Deep Dive. This is where you give us the sources and we dig in to pull out the key knowledge.
Exactly. Quick, thorough, aiming for those aha moments you asked for.
And today we're tackling something visually really arresting.
We definitely are. A listener sent us down the rabbit hole of blood falls in Antarctica.
Yeah.
And we've got a great mix of stuff, a really detailed 2022 scientific paper, but also pieces from NASA, Wikipedia, Earth Sky, IFL Science, even National Geographic. It's quite the collection.
It really is. And the central image, you just can't shake it. This bright, almost shocking red discharge flowing out from the Taylor Glacier. It's just weird against all that white.
Totally. It's called Blood Falls, discovered, what, over 100 years ago now, during the Terra Nova expedition?
Yeah, 1910 to 1913, somewhere in there. It's right at the end of the Taylor Glacier in the McMurdo Dry Valleys. Super remote, super strange.
And it's puzzled scientists ever since. So our mission today, based on everything you sent, is to figure out why it's red.
All right, what's the source of that color? And also, why is it actually important? What's the bigger scientific picture here?
And that 2022 paper, the multi-technique analysis, that's really our anchor. They threw a whole bunch of different scientific tools at it to figure out the mineralogy, the actual stuff making up the surface deposits.
But people were guessing about this long before 2022, weren't they?
Oh, absolutely. The curiosity goes way back. That paper calls it the enigmatic rust color. Researchers were describing it decades ago.
Okay, so what were those early ideas? Where did the investigation start?
Well, back in 1962, you had Hamilton and his team noticing yellowish-brown staining. They identified things like calcite, gypsum, and limonite.
Limonite, that's an iron compound, right?
Yeah.
Hydrated iron oxide.
Exactly. So iron was on the radar pretty early on.
Okay.
Then a few years later, 1965, Black and some others described a reddish-yellow ice cone. Their hypothesis focused on goethite, another iron oxide mineral, as the main culprit for the color.
Goethite, got it.
Yeah, and they also found other stuff mixed in: aragonite, quartz, feldspar, even tiny bits of magnetite and mica.
So these early studies kind of set the stage.
They really did. For a long time, subsequent research sort of referenced back to these initial findings by Hamilton, Black, and later Keyes in the late 70s. It was the accepted wisdom, you could say.
Until this big 2022 study came along and maybe shook things up.
That's the perfect way to put it. They went in with this multi-technique analysis, which basically means using lots of different ways to look at the samples. We're talking FTIR, Raman, VNIR spectroscopy.
Okay. Different ways of looking at how the material interacts with light.
Precisely. And also Mussbauer spectroscopy, which is really good for iron, plus X-ray diffraction, or XRD, to see crystalline structures.
And microscopy, too.
Oh, yeah. SAM-TAM scanning and transmission electron microscopy to get super detailed images, plus EPMA and ICP-OES for precise chemical composition.
Wow, okay. They really brought out the big guns. They weren't messing around.
Not at all. The goal was clear. Get a definitive characterization of the surface stuff, collect it over a couple of trips, and finally settle the question of the red color.
And what did they find? Did they confirm the goethite idea?
Well, this is where it gets really interesting. They used XRD, which is sort of the standard for identifying crystalline minerals. Everyone expected to see strong signals for things like goethite. But the XRD results were underwhelming on that front. They found surprisingly little evidence for well-crystallized iron oxide or hydroxide phases. The expected signatures just weren't really there in abundance.
Hold on. So the main suspect, crystalline iron oxide, wasn't actually the main component causing the color?
It seems not. Now, some of the other techniques, like Mussbauer, did hint at maybe trace amounts of iron hydroxides, things like ferrihydrite, maybe some magamite or hematite.
Okay.
But the results weren't consistent across all the methods. It wasn't a slam dunk. This inconsistency was a big clue that maybe the main source of the color was something different, something not quite crystalline.
Which leads us to
the real star of the show, according to this study: amorphous iron-rich nanospheres, or AFNS for short.
amorphous, meaning they don't have that neat repeating crystal structure.
Exactly right. They're tiny spheres, absolutely packed with iron and, interestingly, chlorine too, but their atoms are arranged in a more disordered way, not a regular crystal lattice.
Ah, so that's why the XRD analysis didn't show strong signals for typical iron minerals, because these nanospheres aren't typically crystalline.
Precisely. XRD is great for crystals, but less so for amorphous or non-crystalline materials. These nanospheres were essentially flying under the radar of that particular technique, at least as the dominant phase.
So how did they nail down that these nanospheres were the culprits then? if XRD wasn't the key.
That's where the electron microscopy, especially TEM, came in clutch. TEM can visualize things at an incredibly small scale.
And I saw these spheres.
They saw them clearly, and the images confirmed they weren't crystalline. Then they used analytical techniques paired with the microscope, like EDX, energy dispersive X-ray spectroscopy, which showed what elements were in the spheres. Lots of iron, as expected, but also significant amounts of silicon, calcium, aluminum, sodium, and, as I mentioned, chlorine was a major component too.
Wow. So it's not just iron oxide, it's this complex little amorphous ball of stuff. How do they actually form and make the water red?
Good question. The thinking is you start with this brine beneath the glacier. It's trapped, very salty, and loaded with dissolved iron in a specific chemical state, F2+ Bay.
Okay, the ferrous state.
Right. Then when this brine manages to seep out at blood falls and hits the air, bam, oxygen.
Oxidation happens.
Instantly. The F2+ iron loses an electron, becomes F3+ or ferric iron, and this process triggers the formation of these tiny disordered nanospheres containing iron, oxygen, hydrogen, essentially iron oxyhydroxide.
And all those other elements get caught up in them too. Silicon, calcium.
It seems like it. They get incorporated into the nanostructure as it forms, probably influencing the exact properties, maybe even the precise color. The TEM images even showed them clumping together with these botryoidal shapes, kind of like microscopic bunches of grapes.
And the chlorine, does the saltiness play a role?
It definitely seems to. Finding chlorine as a major element in the nanospheres highlights the connection to that salty subglacial brine. It's all interconnected.
This is so much more complex than I imagined. It's not just rust, it's nanochemistry happening in Antarctica.
Pretty much.
Yeah.
Which naturally leads to the question, okay, interesting chemistry, but why does it matter?
Yeah, why is this particular red trickle so important scientifically?
Well, blood falls is considered a really valuable extraterrestrial analog study site. Think about it.
Okay.
You got a polar desert climate, super cold, super dry. You've got this unique salt-rich, iron-rich mineralogy. And crucially, you have a microbial community living in that subglacial brine, possibly isolated from the outside world for thousands, maybe millions of years.
Ah, so it's like a potential model for environments on other planets or moons.
Exactly. Places like maybe subsurface oases on Mars, if they exist, or the subsurface oceans we think are on icy moons like Europa or Enceladus.
Yeah.
Could life survive in similar iron-rich, salty, cold, dark places out there? Blood falls gives us a place on Earth to study that possibility.
That's fascinating. So studying blood falls isn't just about Earth, it's practice for looking for life elsewhere.
In a way, yes. Even the practical challenge is getting there. Collecting samples without contamination, dealing with the extreme cold. These are similar to the challenges of robotic planetary exploration, like the Mars sample return missions NASA's planning.
So it's a test bed for methods, too.
Definitely. And finding specific mineral signatures like these nanospheres at blood falls could eventually help us recognize potentially habitable environments from orbit or via rovers on other worlds. If we see similar stuff on Mars, it might point towards a watery, potentially life-supporting subsurface environment.
You mentioned microbes living down there. Tell me more about this hidden ecosystem.
Right beneath the ice, they found this brine. It's incredibly cold, around -7°C, super salty, about 8% sodium chloride, which is much saltier than seawater, and, as we know, very rich in iron.
And there are bacteria living in that.
Yep. They've isolated bacteria, including one called Marinobacter sp, that are adapted to both the cold, psychrophilic, and the salt, halophilic, true extremophiles.
How do they survive?
Their genetics offer clues. They seem to have genes for producing antioxidants, possibly compounds called aeropolyenes, which would help them cope with damaging reactive oxygen molecules, and maybe even genes for pigments like terpenes.
Pigments. Could the microbes themselves be contributing to the color we see at the surface?
It's a possibility, though. The evidence from the 2022 study strongly points to those iron nanospheres as the main cause of the red color. But microbial pigments could, perhaps, play a secondary role or affect the exact shade. It's complex.
And how long have they been down there, isolated?
Estimates vary, but some research suggests the subglacial pool could have been sealed off for, get this, maybe 1.5 to 2 million years.
Wow! A microbial time capsule.
Exactly, which has really cool implications. It connects to the snowball Earth hypothesis, the idea that Earth might have been almost entirely ice-covered in the distant past.
How could life survive that?
Well, places like the Blood Falls subglacial environment suggest that microbial ecosystems could persist, perhaps even thrive, in liquid water pockets within or beneath vast ice sheets. It shows a potential refuge.
This study also seems to highlight how tricky it is to analyze these kinds of samples, right?
Absolutely. That's a major takeaway. The researchers are very clear about the strengths and weaknesses of each analytical technique they used. No single method tells the whole story.
You need a combination.
You really do.
Ah.
They emphasize the need for complementary techniques because each one can have blind spots or biases. For example, they noted how sample handling itself, the process of collecting, freezing, thawing, maybe filtering the samples, can actually change the materials.
Oh, wow. So the act of studying it can alter what you're studying.
It's a real risk, and it's a critical lesson for analyzing precious return samples from, say, Mars or an asteroid. We need to understand how our methods might inadvertently change the very thing we want to learn about.
Did they run into other analytical challenges?
Yes. They mentioned that with XRD, it was hard to definitively sort out complex mixtures, especially when you have very fine-grained stuff mixed with amorphous material, which is exactly what they found at blood falls. And that's relevant for interpreting rover data from Mars, too, which often deals with complex mineral mixtures.
Because the main coloring agent, the AFNS, was amorphous.
Right. Standard techniques that rely on finding crystalline structures weren't sufficient on their own. They needed that multi-pronged approach.
Did they notice if the nanospheres changed over time after being collected?
Interestingly, yes. The nanospheres seemed pretty stable in samples collected in 2018 and kept frozen. But they appeared less common, or maybe different, in older samples collected back in 2006.
Any idea why?
They suggest a couple of possibilities. Maybe the nanospheres slowly change over time, even when frozen, a process called ripening. Or perhaps it reflects differences in the brine outflow or the exact timing of sample collection between those years. It's another complexity.
Thinking about these nanospheres, does this research connect to other work on nanoparticles, like in medicine or material science?
It does, indirectly. The paper briefly mentions the huge field of iron oxide nanoparticle research. These engineered nanoparticles are used for all sorts of things in biomedicine: contrast agents for MRI scans, targeted drug delivery, even heating up tumors in magnetic hyperthermia therapy.
So understanding naturally formed iron nanospheres adds to the knowledge base.
Exactly. While the blood falls nanospheres are natural and amorphous, studying their formation, properties, and stability contributes to the broader understanding of how iron oxides behave at the nanoscale, which is relevant across different scientific fields.
Okay, let's bring it back to space. How does figuring out the source of the red color at blood falls help in the search for life beyond Earth, specifically using things like orbiters or rovers?
It's fundamental. We need to know what surface features, what mineral signatures might indicate a habitable environment hidden below. If we see certain iron minerals or perhaps amorphous signatures like these nanospheres on Mars' surface, could that signal a subsurface brine?
A signpost for where to look closer?
Precisely. The study showed that the spectral data they got from blood falls using techniques like V and IR matches the kind of data remote sensing instruments collect and clearly indicates ferric iron. And as several sources mention, the McMurdo Dry Valleys are one of our best Mars analogs on Earth.
Which is why NASA is so interested.
Absolutely. NASA keenly studies extremophiles like the blood falls microbes because they represent potential models for life that might exist or could have once existed under the harsh conditions found on other planets and moons.
Okay, let's try to wrap this deep dive up. What are the absolute key takeaways from puzzling over blood falls?
I think the biggest one is the identity of the red coloring agent. It's not primarily crystalline rust like goethite as we thought for decades. It's these amorphous iron-rich nanospheres formed when the subglacial brine hits the air. That's a major revision.
And that discovery really highlights the need for using multiple scientific tools.
Definitely. It's just you can't rely on just one method. Especially for complex, potentially exotic materials, you need that complementary toolkit to get the full picture. This is super relevant for analyzing samples from other planets.
Beyond the color, the significance lies in blood falls as a whole system, right?
Exactly. It's this incredible natural laboratory. It gives us insights into how life can survive in extreme, cold, salty, dark, iron-rich environments for potentially millions of years.
Sure.
That has implications for Earth's past, like Snowball Earth.
And for the search for life today.
And absolutely for the search for life today, particularly on icy worlds like Mars' subsurface or moons like Europa. It shows us that seemingly barren, icy landscapes might hide active, albeit extreme, ecosystems.
It really does leave you thinking. If an environment this unique can be hidden beneath Antarctic ice, what other bizarre and wonderful pockets of life or unique geological processes are waiting to be discovered in extreme places here on Earth?
Or elsewhere. It definitely sparks the imagination about what might be hidden under the Martian surface or within those distant ocean moons.
A great thought to end on. The universe might be stranger and more full of surprises than we think.
Always worth remembering.
Well, thanks for digging into blood falls with us. We hope you found that as fascinating as we did. Join us next time for another deep dive.